Herewith, I open this academic session in which Lieve van Veggel will defend her academic thesis, All You Can Eat 3, Restoring Myelin, the Brain's Favorite Fat. Zoals u hoort, mensen in de zaal, is de sessie vandaag in het Engels, omdat we veel opponenten uit het buitenland hebben. Dus u zult her en der wat moeten vertalen onderling. Ik doe dat een beetje zachtjes, want anders kunnen we Lieve niet verstaan. Dus ik wens u nu heel veel succes. Good luck with your defense, and you can start with your talk.
Thank you, Pro-Rector. Dear members of the Corona, dear colleagues, leveren and familie. Today, I would like to talk to you about my project, All You Can Eat 3, Restoring Myelin, the Brain's Favorite Fat. But before I jump into the main results of my study, I need to give you some more information about the foundation of my work. So first, as I said, myelin, the brain's favorite fat. But what is myelin exactly? Well, as you can see here, myelin is located around the arms of nerve cells, also called neurons. Myelin is produced by cells called oligodendrocytes, and they basically wrap around these neurons. And therefore, myelin has two important functions. First, it plays an important role in protecting these neurons against any outside damage. It also plays an important role in the communication between neurons. As you can see here, the signal that neurons communicate with can jump these pieces of myelin, making the communication more efficient and much faster. The situation I just explained to you is what happens in the individual with a healthy brain. But what I'm interested in is to see what happens to myelin in people diagnosed with multiple sclerosis, or MS. Here we know that immune cells come in and start attacking these myelin-producing oligodendrocytes. This leads to the loss of myelin, which we call demyelination. But what does this mean for patients? Well, since MS is an autoimmune disease which can affect patients in different ways, they can also experience very different symptoms. This is not only limited to muscle weakness or trouble with balance, which most people associate with MS, but also things like bladder issues, heart problems, trouble speaking, losing eyesight, or also importantly, trouble with memory and cognition. For most patients, these symptoms come and go in the early phase of the disease, as you can see in this graph. But at a certain point, these symptoms only get worse. This is what we call the progressive stage of the disease. For these patients, no proper treatments are currently available. There are also no treatments which can delay the progression of the disease. But now, let's look back what happens after demyelination and why patients with MS can still recover in the early phase of the disease. So fortunately, your brain is normally able to repair any damage to myelin by producing new myelin. This process is shown here, where oligodendrocyte precursor cells, or as I like to call them, baby oligodendrocytes, can grow up and become mature myelinating oligodendrocytes. This process is called remyelination. However, in the brains of patients with MS who have entered the progressive stage, these baby oligodendrocytes no longer want to grow up and take part in myelin repair. Now, on to the next part of my title, All You Can Eat 3. EAT3 is a protein that can transport molecules from the outside of the cell to the inside of the cell. And interestingly, EAT3 is located on oligodendrocytes as well as these baby oligodendrocytes. But what does EAT3 actually transport? Well, it can transport two different molecules into the cell. Once these molecules are transported, together with a third part, they can form building blocks and become a larger structure which can function as a shield. This shield can protect these cells against any outside attacks. This was interesting to us, so we decided to study the role of E3 in these oligodendrocytes and its potential to push these cells to grow up again and become mature myelinating oligodendrocytes. So what did we actually study? Well, first, we were interested to see what would happen if we would increase E3. We think that this will lead to more shield formation and thus better protection of these oligodendrocytes, meaning that when an immune cell comes in and releases their attack, the attack can be fought off and the cells will survive. On the other hand, we were also interested to see what would happen if we would block E3. We think this would lower the amount of shields and therefore lower the protection of these cells, meaning that when these immune cells come in and release their attack, the cells cannot fight it off and they will become damaged. So overall, we expect that more E3 can improve the baby oligodendrocytes growing up and becoming mature myelinating oligodendrocytes because they can defend themselves. But I also need to explain to you how we looked into this. We were interested in E3 modulation within the context of multiple sclerosis. Therefore, we needed a proper model to study this. First, we needed access to normal, healthy mice eating normal food and normal myelination in their brain. Secondly, we mimicked MS in mice by giving them a myelin-damaging diet, which we know leads to the demyelination of their neurons, so myelin loss, mimicking MS. Of course, we also have to be able to measure myelin to make sure that the effects we are expecting are also occurring. We did this by using something called visual evoked potentials, or VEPs. How does this work? Well, we first put a mouse to sleep, then we place an electrode in the back of its head where the visual cortex is located, and then we put a light source in front of the eyes of the mouse. We briefly flash this light, the eyes register this, and an electrical signal travels to the back of the brain. We know when the light flashed and when the signal arrived in the back of the brain. If there is proper myelination, then we know the signal travels very fast. But what then happens in mice where we mimic MS? Well, there we know that there is demyelination in their brain. So if we would do the same test again, we flash the light, we expect that the signal takes much longer to travel to the back of the brain. Okay, so what did we actually see in our experiments? First, we looked into what happens if we increase E3. As a little recap, here we expected that this would increase the protection of these cells and allow them to become mature myelinating oligodendrocytes. So first, in our healthy mice, we indeed see that the signal travels very fast, as we expected. Then in our mice where we mimicked MS, we see the signal indeed takes much longer to travel to the back of the brain, also as we expected. But then, strangely enough, in our mice with increased E3, we see that the signal takes even longer to travel to the back of the brain, meaning that there are signs of demyelination, less myelin. Then, of course, we were also interested to see what would happen if we would block E3. And here we also see the opposite of what we expected. So here we see much faster signaling, so signs of more myelination, remyelination. So this was interesting to us, and we decided to move closer to the mechanism of E3 and look into the levels of shields. So first we looked into our healthy mice. There we see very low levels of shields, as expected, because there's very little attacks happening in their brains. Then in our mice where we mimicked MS, we see that the shields slightly increase. But then when we increase E3, we see a dramatic increase in the amount of shields, as expected. And then when we block E3, we see that these amounts of shields are lowered again. However, not less than in our mice with MS and normal E3. So we've gathered some interesting results. So as a little recap, we looked into increasing and blocking E3. We expected that increasing E3 would lead to enhanced protection of these cells, meaning that more baby oligodendrocytes can grow up and take part in myelin repair. However, we see the opposite, where increasing E3 leads to more demyelination. On the other hand, we expected that blocking E3 would lead to less protection, so less myelin, but here we also see the opposite, where blocking E3 leads to more remyelination. When it comes to the amount of shields, we do see that increasing E3 leads to increased amounts of shields, and blocking E3 leads to lower amounts of shields. So we believe that our findings regarding the role of E3 in oligodendrocytes is only the tip of the iceberg of what there is to discover. As to why we see the opposite effect of what we expected, I can merely speculate about that here today. But one interesting thought could be that E3 is simply inserted in the opposite direction in these oligodendrocytes. However, this does not fully explain why we still see increased in shields when we increase E3. A second interesting idea could be that the protection that we see due to increasing E3 is simply still not enough to fight off these immune attacks. And a final interesting thought could be that E3 heavily favors the transport of one of these building blocks into the cell. Nonetheless, my research has only uncovered the tip of the iceberg of what there is to discover when it comes to the role of E3 in oligodendrocytes within the context of multiple sclerosis. So what I would like you to remember from today is, first of all, there is a very big need for therapies that focus on restoring myelin. People diagnosed with progressive MS currently have no proper treatments available to them. Secondly, I've shown you that E3 is an interesting therapeutic target. However, not as expected, because I've shown you that blocking E3 can improve myelination and needs to be researched further. What I've shown you here today is a complicated scientific story, but that is what science is. Sometimes the data you gather leads you down an unexpected road, but what matters the most is that it's interesting and it adds to the world of science. So, to conclude, what I've shown you here today is only a small part of the work in my thesis. I've mainly talked to you about Chapter 4, where I've looked into increasing and blocking E3 and its effect on myelin. But in Chapter 2, I've also looked into the use of a tool compound that can activate E3, highlighting the importance of reproducibility of data. In Chapter 2, I dove into recent literature looking at oligodendrocytes and their role in different neurodegenerative diseases, highlighting their potential as therapeutic targets. And finally, in Chapter 5, I look back at this challenging project in the form of a perspective. Here, I've identified pitfalls, but also positive lessons learned. And I hope this can help researchers in the future to generate new research questions when it comes to the role of E3 in oligodendrocytes. All in all, I hope I gave you an insight in the work that I've done these past 4 years, and you learned something new today. Thank you very much for your attention, and I now give the word back to the prorector.
Thank you very much for your very clear presentation with interesting findings. I'm almost sure that people will have questions on these findings. That's why we're all here. But before we start, I would like to introduce to the audience the 4 people who are not allowed to speak today, because they are the people who guided her through the thesis and the research. And those are her supervisors, Tim van Mierlo and Rudy Schreiber, and the co-supervisors, Professor Hellings and Dr. Delian. Welcome, glad to have you here. This is a double promotion, so we're very proud that the people from the University of Hasselt are here as well. And we will now start with the questions. Are you ready?
Yes.
We are too. That's always good to know. I give the floor first to Professor Van den Hove, who is a professor in neuroepigenetics at Maastricht University. He's also chair of the assessment committee, and in this committee today also the secretary. So quite a lot of functions. The floor is yours.
Thank you very much. Dear candidates, first of all, my sincere congratulations with this very nice thesis. It was a pleasure to read. Also, my compliments for your presentation today. Very clear and helpful for the audience, I think. Also, my compliments to your promotion team. So, a superb job. Before I dive into a few questions I have, I wanted to ask one of your panelists to read out statement number 12. If possible, because she's not local, I think, but anyway.
Wie deks wordt ze niet prijsgegeven, maar heel zijn kroon toch opgericht.
Well done, thank you very much. Well, me being local, I hear this statement every other week with my kids in a soccer stadium. And we get defeated quite a bit because this statement is about defeat. So it sort of says, how often have you, as a city, because it's from the Maastricht anthem, not been defeated, but you still kept your head up high? I think this also translates nicely to science, because science is all about failure and defeat. And I think you did a very good job in overcoming that. My minor comment, before I go to my questions, I think this should have deserved statement number 11. Us being local, number 11 is something special, so I think you should have switched them around. Anyway, let's move on to my first question, which is actually on the model used. So you talk about the Cooperzone model, you talk about differentiation block, etc., and then you have your strategy to try to move your way around and see whether this can have a beneficial effect. But I was wondering to which extent this model is actually modeling a differentiation block. So maybe you could briefly explain a bit more about the model and how you see that in light of myelination and the myelination block.
Highly esteemed opponent, thank you very much for your kind words, and I do agree that it should have been statement number 11, missed opportunity. Thank you very much for your nice question. Let me start by saying that there are no perfect MS mouse models, far from it. And indeed, the Cooperzone model, it's a copper chelator, and it basically kills off your oligodendrocytes. And then a new wave of OPCs come in, and if you give Cooperzone long enough, it will also kill those new OPCs. So it's definitely not the same principle as we see in MS patients, where there is indeed the OPCs are there, but they somehow don't want to differentiate. Here we just kill them off in the Cooperzone model and they don't get the chance. They only get the chance once we stop the diet. So I fully agree with you that it doesn't perfectly mimic the situation in MS patients. However, we do not have proper means as of yet to mimic this in a better way. So I think this is the best model as of yet to really look into de- or remyelination effects.
Okay, thank you very much. Then my second question would actually focus particularly on figures 8 and 9 of chapter number 4. And we don't have to dive into the figures specifically, but if you look carefully, you do see a discrepancy between the VEPs, so the evoked potentials, and actually your analysis on myelination, particularly dealing with the MBP staining. So, for example, you can see that both compounds, they actually work, and I'm talking now about inhibition, that both compounds targeting E3 and inhibiting them, they do seem to work at 6 weeks plus 4 days. However, only PDC seems to improve myelination by itself, and while both compounds work at the level of VEPs. But if you then look at PDC, it actually already works at 3 weeks in terms of restoring myelination. So how should I see this discrepancy between the effects that your compounds have on MBP and how that translates into the VEPs or doesn't translate? What's your view on this?
Thank you for your question. You're definitely measuring something different with both measures, right? So one is MBP staining at a very different location as where you look at with your VEPs. So with your VEPs, you look at the entire optic nerve, and we know that probably the demyelination effect that we see there is more located towards the visual cortex, which is nowhere near the region where you're looking with your MBP staining. And MBP staining, it's very much a single time point that you're looking at. So you might also not be looking at the right time frame. There is also quite some variation, as you can see in these stainings. So I believe more in these functional measures, like the VEPs, and go off from those measures, and then use these stainings more as to support the data that you see there. But I agree with you, there are definitely discrepancies. So we cannot for certain say, okay, at 3 weeks is where the effect happens, at 6 weeks or at 6 weeks and 4 days. Yeah. So it's difficult, and ideally I would have done more measures into myelination. We're still working on TEM images, so we hope that that can actually support our VEP data as well.
Okay, thank you very much. Yeah, you talked a bit about variability in your data, and I also was triggered by that. Of course, this is something usually negative, but it could also be seen as positive in this respect, because did you try to correlate the measures at the level of MBP, for example, with your VEPs, in the sense that those that score low also show aberrant behavior and the other, and vice versa?
Yeah, I've been thinking about this, but I have not, also because the animals that we did the VEP measurements with are not the same animals, for example, that we did the MBP staining at 3 weeks. Because I cannot measure.
Only the later ones.
Yes, and I did not do VEP measurements on those animals. So I've always tried, for as far as possible, to take the same animals for every VEP measurement, so 6, and the other 6 were then sacrificed at the 3-week time point. So that for that time point, not possible. But I think it would be interesting to do it for the later time points, for sure, to see also maybe non-responders, responders, sort of, to our inhibitor, for sure. Yeah.
All right, given I think the limited time and then other opponents, I'm very happy with the answers. Give the word back to the prorector. Thank you.
Thank you very much, Professor van den Hove. Then the floor is for Professor Rigo. He's a professor of neurobiology from Hasselt University. Welcome to Maastricht. And the floor is yours.
Thank you very much. I would like to join for my felicitations for the work you have written and the very beautiful presentation. I mean, you made also drug development as an important scientific advancement as such. It doesn't happen so often, so it's really refreshing to read that. And well, this made, in particular, the impact section of your book very, very good. And so I have 3 main questions, and those are very general questions. And the first one is very challenging because I will challenge the order of the chapters that you choose, actually. You begin with a very interesting hypothesis, and you nicely show here that the hypothesis was not correct, and it was the inverse. Why didn't you begin with the in vivo experiments to see if it's blocking or not blocking who is doing something?
Highly esteemed opponent, thank you very much for your kind words and interesting question. We wanted to start the in vivo basically right from the get-go when I started this project. But the in vivo experiments have been very challenging. We had initially a very different setup that we wanted to use. We wanted to use Cree mice, and then the mice that Professor Moya has in Chile to create overexpression in these specific OLIC2 Cree mice. But we went to various kind of iterations of our in vivo plan. So I get your question. We definitely wanted to do it as early as possible, but it was just due to practical reasons, sadly not possible. But yeah, it would have been better. I agree with you for sure.
Okay, very good answer, I think, and very honest answer. Well, linked to that, you decided to compare overexpression with pharmacological inhibition. So I understand at that moment there was no way to have a pharmacological stimulation of the system. But why not to have chosen for a knockdown system in a certain way to compare?
Yeah, so we first indeed started with the AAV overexpression model and wanted to see what was happening there. And we did think indeed about knockout and also CRISPR in vitro. But then recently a paper came out that showed that PDC inhibition is indeed beneficial in the context of white matter damage. So we thought it was only fitting to actually use that same compound along with the other inhibitor that we had been using because there seemed to be some therapeutic potential in E3 inhibition. And a knockdown or a knockout system, I think, is very valuable, but it's not translatable to a human setting. So that's why we aimed to go for the most translatable setup, which in my view is a pharmacological approach, but it comes with its challenges for sure, because you're less specific, of course. So yeah, that's the kind of reasoning behind it for me.
Okay, and do you know how specific those inhibitors, so the developed inhibitors are?
So PDC is more a general EAT inhibitor. It also targets EAT1 and EAT2. So that's definitely not as specific as it should be. But since they use it in the other study, we thought it would be good to also take it along. Inhibitor 3A has only been described in one in vitro paper, and they claim it's the most specific EAT3 inhibitor out there. So yeah, I think 30-fold. I don't entirely know the numbers, but indeed more specific than TFBT-BOA or all the other EAT inhibitors that are out there. So it should be fairly specific, but if I would do a follow-up experiment after this PhD, I would definitely be looking into this compound because it's very interesting, the results, but it needs to be characterized further, for sure.
Mm-hmm. Then another question is about oxidative stress that leads to OPC dysfunction in some individuals, but not in others. Why is it so? Because, I mean, if this is the exposition to oxidative stress as such that induces the OPC dysfunction in any individual, or is it actually the diminution of protective mechanisms by some individuals that renders them more susceptible to this oxidative stress? What is your meaning about, well?
Yes, if I understand correctly, you're asking why there are differences between oxidative stress kind of susceptibility, as to say. I think that is because the kind of oxidative stress antioxidant defense mechanism, it's a very complicated system. As you can read in chapter 2, the review, there is so many different factors that come along with antioxidant defense, and in basis, oxidative stress is a good thing, right? Very limited levels of raw species are necessary for proper cell signaling. And also, as you've read, OPCs have very limited antioxidant defense, but also this can vary between people, between mice, a lot, as I can imagine. So I think there is a lot of variety there because there are so many players involved in this complex system. So I think it's definitely not a clear-cut, okay, this amount of oxidative stress is too much, for sure, I think, yeah.
Okay, and a very last small question. But in the introduction, you talk about baclofen acting on spasticity. I'm curious about that because I didn't know that baclofen has an effect on spasticity.
I'm not super familiar with this compound, but yeah, I did find this in an interesting paper where they try to have like a different approach to MS treatment and not the current kind of status quo where you go from not a super harmful compound and start from there and then move on. This is from a paper where they start giving these more harsher treatments and indeed also these symptomatic treatments early on, and then indeed they see these type of effects. So if I remember correctly, it's not like a distinct one-off effect from this compound, but if it's taken together with other treatments for MS, then the effect is there.
Thank you very much for the nice discussion.
Thank you very much, Professor Rigo. Then we go on to one of the opponents online. And you take a sip of water because you do a lot of talking now. We go to Dr. Gasparini. He's the Executive Director and Head of Neuroinflammation of Novartis in Basel, Switzerland. Welcome to this session and thank you for being here. And the floor is yours.
Thank you, Co-Rector. Dear Lieve, congratulations first to your performance today in the presentations and the first sessions in answering questions. I would like also to thank as well your supervisor who is actually guiding you in this very difficult challenge that you rightfully illustrate in your work in the sense that actually identify the target, validating it, acting on animals with different compounds, and potentially verifying or contracting an hypothesis is a very challenging word. And actually, I'm happy to see that actually you described actually very well the difficulties that not only in academical, but most likely in the pharma industry is present, and it's our work every day. Having said that, I have a couple of questions, dear Lieve, and one is starting with your introductions. You had a nice introduction about the disease, the therapeutic approach to the disease, the actually lack of absolute therapeutic solutions for contracting the progression of the diseases. And then from then you went down. You identified a cellular mechanism related to OPCs, and ultimately you made an hypothesis, which is a cellular or molecular hypothesis, by activating this E3 receptor. Having said that, you rightfully said that there are approaches to contract remyelinations in the context of MS, and you gave some examples. To your knowledge, actually, what were the motivations for these investigational therapies to go for remyelination in MS? Just a couple of words.
Esteemed opponent, thank you very much for your kind words and your nice question. If I understand correctly, you're asking why, like what the motivation is of people to try to find remyelinating therapies?
You specifically cited examples, clemastine and metformin being investigated in MS. Remyelinations.
Yeah.
What was the rationale to use those agents for going into remyelinations potential therapies?
Yeah, so for example, metformin is, I think, a very interesting one because it's known to play a very important role in kind of turning back aging, right? So if it's given to aged OPCs, which is gaining a lot of attention nowadays, that is one of the problems in the context of MS, then you can make these OPCs young again. So that's very, I think, very interesting, and they can then participate better in myelination. Yeah, metformin is also an interesting, complex downstream mechanism, which also, if you look far enough, also comes back to also oxidative stress. It's also mentioned in the review, I believe. It's a very interesting new compound, but it's also hitting some bumps in the road in the development, but still gaining a lot of traction. Can you remind me of the other compound you mentioned?
It was clemastine, but I think it's probably the same rationale. So let me go to the other question that I have here, and it's about the use again of the cuprizone model, as you mentioned. Cuprizone is a copper chelator inducing active death of oligodendrocytes, and you analyze it rightfully using a less invasive actions of your molecules using visual evoked potential. But you look at it from one single point in time, whereas actually maybe a way to better understand how this remyelination takes place is to see differentiation, how fast they remyelinate the oligos and how fast they come back. Because actually in the rodent model, oligodendrocytes, when they are depleted, OPC will repopulate, migrate to the right place, and do their job, honestly. So eventually, again, looking at the different time points would allow you to see how the difference in how fast this remyelinates. Have you thought about that?
Yeah, I think that's a very interesting aspect. So now we've very carefully tried to select our time points because we were not sure if the effect would be a demyelination or a remyelination effect. That's why we chose 3 weeks, where there is a very big demyelination wave, 6 weeks at the end of basically the most demyelination, and then 4 days after we stop cuprizone diet, because we know from previous studies that that is the time point where you can pick up remyelination. But I do agree that it would be interesting to test these mice, every day if possible. But VEP measurements is fairly uninvasive, but still you have to sedate your mouse. Some of them don't come out of it, and we didn't want to risk losing these mice and not getting to proper sample size. But I do agree that it would be interesting to look 1 day after cuprizone, after we stop cuprizone, and 2 days and 3 days as well, to see indeed how fast this is, because we do expect that there's also timing differences, right? We see with the AAV that if we overexpress E3, it could be no remyelination, but it could also be delayed remyelination. Now, we didn't look further than these 4 days, but if we may have looked at a week after stop cuprizone, then we might have seen still remyelination. So I think it's a very valid suggestion.
Thank you. My time is going short, but I have a last one, and it's related to a second model.
Dr. Gaspari, I'm sorry, yes? But when you're having fun, time flies, and we almost run out of time.
Okay.
So thank you very much and for your
You're
questions. Welcome. And we go to the next one, and people in the audience pay attention. Now we're going from Switzerland to Scotland. Dr. Gloucester, she's an associate professor in molecular biology at Andrews University in Scotland. Dr. Are you here? Yeah.
I am indeed. Can you hear me?
Yes.
Thanks very much for a wonderful presentation, and it was of great interest to read your thesis and hear about all the work that you've been doing during your time in your PhD, and I think it's an impressive body of work that you've managed to produce. So I have a few questions, and the first one is in relation to the chapter 5 on the drug discovery pharmacological interventions, and you mentioned in there the idea of using structure-based drug discovery at the first part of your as part of the original plan of your PhD. And obviously a lot has moved on in the time during your PhD in structural biology, AI. And so I was wondering if you could maybe outline how you would start that project now in the context of the experimental and computational techniques that have been developed in the last 4 or 5 years.
Esteemed opponent, thank you very much for your kind words and your very relevant question, I think. So yeah, indeed, when we started, we did want to take a structure-based drug discovery approach. And when we wrote this project, there were no structures available for E3 as of yet. Now, I think I looked yesterday, it's 14 of them already in the past 4 years in different states and with different substrates in there as well. So I think that has already happened. That's already a major change, I would say. As to the use of AI, I also attempted molecular docking myself, which proved challenging. I know we discussed this as well. But right now there's a variety of different tools. AlphaFold, for example, is very interesting. I think there is still no E2 structure published, if I'm correct, so that their AlphaFold could be of use. And definitely also with the docking, I'm sure these DocVina, all of these programs can improve immensely from AI input. So I would be excited to actually start this again. I'm very, a bit sad that we couldn't actually get to this within my project. But I think there are so many different paths you can take with AI nowadays, and it would be very interesting to investigate, yes.
Yeah, no, I agree. Thanks. Thank you. So then I want to talk a bit about the work in chapter 4. So in terms of the inhibition of E3, I was wondering if you could comment on whether the inhibition could be related to the transport of cysteine versus glutamate, and if there's any information on the relative affinities of the inhibitors that you've got versus cysteine and glutamate. Would it be blocking the function of transport of both, or do you think there would be a way to delineate if there was preferential inhibition of transport of one amino acid over the other?
So I would like to focus on inhibitor 3A because that's the more specific one for E3. There's not that much known about this compound. We don't know if it's a competitive inhibitor. I expect not, because it doesn't mimic an amino acid-like structure like glutamate or cysteine, which most of the other inhibitors to EATs do. So they're competitive inhibitors. So I expect it's allosteric somewhere, but we don't know where it would bind. In terms of affinity for either one of the two substrates, we know that E3 favors transporting glutamate over cysteine, because transporting cysteine is a little more complicated because it needs to be deprotonated in order to be transported. But in terms of the compounds inhibiting either one more than the other, that's not known. So that would be very interesting to see and could explain potentially also why we see the effects that we see, that inhibition is actually positive. Maybe it inhibits glutamate transport more. So yeah, I think it would be interesting, and we need to investigate that compound. Like I said earlier, the compound needs to be characterized more and definitely looked into detail what it does actually, yeah.
And following on from that, really, how did you decide the dose of the compounds that you were going to give to the animals? So I think it was different between the different inhibitors that you used. How did you come to those dosing regimes?
We based on the dosing of the other study that's recently been published, where they also looked at white matter damage and then gave PDC to their mice, but they directly infused it into the brains of the mice. And now we went for subcutaneous injection, which as far as my knowledge goes, has not been attempted with this compound, but we try to kind of mimic the same concentration. So we try to recalculate and use that same concentration. For inhibitor 3A, it was also basically a guess. So we went for 2 different concentrations in our study where we studied brain penetration. And we saw, maybe not surprisingly, that the highest concentration leads to the highest concentration in the brain. And we didn't see any adverse effects in our mice, no strange behavior that could be signs of like off-target effects or something. So we decided to just go for the highest dose. And based on other studies, what we've done, so that's the decision making there.
And you touched a little bit on the specificity of the inhibitors, but can you be sure that you're not actually getting some inhibition or activation even of EAT1 and EAT2 during these animal experiments? How convinced are you that the effect that you're seeing is down to EAT3 alone?
We cannot be certain. For PDC, it's known that it's not EAT3 alone. It's definitely also EAT1 and EAT2. For inhibitor 3A, we can only go based off that one paper where they say it's specific. But I think because our effect in our overexpression model is oligodendrocyte specific, because there we have overexpression specifically in MAC cells, and we see the opposite effect in our inhibition model, I would say there is a strong link, definitely, that we're hitting oligodendrocytes. But it's, definitely not a given. We know that EAT1 and EAT2 are expressed in astrocytes and in neurons, respectively. So it might well be also that we have some effects that are perhaps neuronal as well. But at least it's a nice effect that we see, but it needs to be researched further, for sure.
Yeah.
Thank
I have
you,
one more question.
Dr.
I don't know if there's time. Yeah.
Thank you, Dr. Kloster. Very much appreciated. Okay. Then the floor is for Professor Heitman. She is a professor of molecular pharmacology from Leiden University. Welcome to Maastricht.
Yeah. Thank you very much, Rector. Dear candidate, first of all, of course, I also want to congratulate you and your promotion team with this beautiful thesis. I've really enjoyed it a lot, also from a more personal perspective, because multiple sclerosis runs in the family, and it was actually the reason why I started on this career path many years ago. As you know, I've collaborated on E3, but I'm actually more of a G protein coupled receptor expert, and then my expertise lies in vitro drug discovery. So I have a little bit of questions in that area. But I first want to start with your beautiful cover. We had some contact about it via email, and you said that AI was of help here. But I see a woman alone in a dark forest. How does this link to your PhD project?
Highly esteemed opponent, thank you very much for your kind words. An interesting question that I've gotten a lot these last couple of weeks. So it's actually a woman that comes out of the dark forest and moves into the light, where all of her friends and colleagues and people are. So it's a positive turn on this project that was a roller coaster, but it all turns out fine in the end.
Okay. Yeah, I had indeed noticed the light as well, but it didn't. So, in chapter 1, you clearly explain the hypothesis why E3 can be a novel remyelination target, and that it's one of the amino acids that it transports is cysteine. And do you know if the expression levels of E3 or any somatic mutations play a role in the development of MS?
Not as far as I know. So E3 has not been researched in the context of MS. I think we are the first to actually do so. In terms of expression studies, it's been very limited. There are some signs in OCD and depression, but not in the context of MS. And we also did not pick it up in one of our own studies where we did an EWA study looking at normal appearing white matter and lesions of MS patients. Yeah. It also did not pop up there. Okay. So as far as we know, it's not necessarily affected in MS. One study that has been published is a small study where they gave MS patients interferon beta. they saw that E3 expression decreased. So they do also mention that in the recent paper with the white matter hypoperfusion model, but yeah, that's all that's out there, basically.
Yeah, and a similar question is about the levels of cysteine that is being produced in MS patients. Do you know if that is different and in healthy people?
As far as I know, also not just the general consensus that OPCs have very limited antioxidant defense mechanism, so that they have, in general, very low glutathione levels, and that also alongside aging and in the context of different diseases, that doesn't help, of course.
Yeah.
It gets diminished even further, especially because they get more and more ROS attacks. But as far as I know, specific MS studies in the context of E3 and cysteine are very limited.
Yeah, because a quick Google search taught me that they are now doing a clinical study where they treat MS patients with cysteine.
Yeah, and cysteine.
So what about targeting the protein that is involved in the conversion of glutathione to cysteine? Would that be an interesting drug target? And how would you start such a drug discovery project?
Yeah, I think for sure. So N-acetylcysteine is an interesting one because it shows a bit conflicting results. I think it shows some positive results when it comes to cerebral blood flow examples, but then not for a fatigue. But it shows promise. But there are definitely different targets that we can look into. Cysteine is first coupled to glutamate, and then it's formed into glutathione eventually. But what I like about E3 is that it's fairly oligodendrocyte specific. Of course, glutathione synthesis is very important all over the body, and the synthesis is very, well maintained. And these enzymes that are involved are, as far as I know, very similar in different organs.
Yeah. So you would not favor that approach?
In terms of specificity, I would say no, but it's an interesting thought, I think, because N-acetylcysteine has been shown to be an interesting therapy as well, so it's definitely something to think about. I agree.
Yeah, okay. I'm looking at the rector, but I think I still have time. Yeah. So, in chapter 4, you use an in vivo model. I'm, of course, not an expert in this area, and you need to help me. So this AAV transduction that you use, do you then upregulate the human E3 or the mouse E3? Mouse, okay. So then I had a question. These inhibitors, I think they were developed for the human E3. So what do you know about species selectivity or aselectivity between the human and the mouse protein?
I know that mouse and human E3 is very homologous. As far as I know, it's very similar. The two proteins are similar to each other. Also, its transportation is fairly similar. As far as I understand.
Probably the overall homology, and maybe not focused on the binding site of these ligands, which are not known.
That's true. And we also don't know if our inhibitor 3A, so this has been only studied in an in vitro context. If I remember correctly, it was HEK cells, so it's also not a mouse-derived cell. So we were unsure if this would work. It was kind of a guess, I think, to use it in vivo, but we do see that it has an effect. But if it's then E3 specific, I think that's a fair question, and we need to look into that for sure, also for PDC.
Okay, thank you very much. I give the word back to the rector.
Thank you, Professor Heitman. Then we continue the opposition with Professor Moya. He's a professor in physiology at the University of Valparaiso. So people in the audience, pay attention, we're now in South America. Let's see if we can get Professor Moya on screen.
I think he left.
Can you hear me?
Yeah. Yes.
First, congratulations, Lieve, for this fantastic presentation today, and also for the impressive progression over this complex path. I've been following your progress steady over the last year. So from the distance, I feel very proud of seeing you and hearing you today. I have some very general questions. We have discussed your results several times. Could you please share your the perspective from the genetics of multiple sclerosis? It is known that multiple sclerosis is a a disease with complex genetics, but the genetic contribution is established, is clear from both twin studies and family studies. So can you remind us if there is any suggestion or link to the SLC1A1 genomic loci from the linkage or candidate studies?
Esteemed opponent, thank you very much for your kind words and your interesting question. I've tried to dive into this also these last couple of weeks in terms of genetic studies related to E3 in general. I think in the context of MS, as I mentioned before, there is very little known. Again, as far as I know, we are the first to look into E3 as a therapeutic target in the context of MS. It also did not pop up in earlier EWAS and GWAS studies. And yeah, as you know from your own work, it has been linked to OCD and also depression. But in terms of genetic studies into MS, I'm fairly certain that there is no clear link with E3 apart from what I've just mentioned, the small study where they do see a decrease in E3 expression upon interferon beta treatment. But as far as I know, there is no clear differences in genetics when it comes to E3 between patients with MS and healthy individuals.
Thank you. So my second question is, if you were able to generate a selective allosteric modulator targeting E3, how could you deal with the tissue specificity, provided that this transporter is also expressed peripherally, for example, in kidneys?
I think it's almost impossible. If you design an allosteric, let's call it activator in this case for E3, it's very difficult, of course, to make that cell specific because indeed E3 is present in other cells. So it would be a matter of studying the correct dose, I would say, and really dive into those expression profiles and also look at administration routes, I would say. If you're targeting in the brain, of course, it needs to penetrate the blood-brain barrier, et cetera, et cetera. So I think those are definitely characteristics of molecules that you can play with that make sure that it gets to the right organ. But in terms of just in general designing it, I think it's a challenge, I would say, as it was a challenge when we tried to make an activator that would only activate cysteine transport, as you remember, and not just glutamate. But I think it's definitely interesting, and it's something, as I'm aware, is not done as of yet in current therapies on the market, but nothing is impossible, I think.
Excellent. Do you think that the multiple substrates being carried by these transporters could explain, at least in some part, some of your unexpected results? So I'm thinking about glutamate versus cysteine. Perhaps.
So we know, of course, that E3 also plays a very important role in counteracting glutamate excitotoxicity. E2 is definitely the big player there and the most important one, but E3 also for sure plays a role in oligodendrocytes. So I think that's definitely something we have to keep in mind. We're not looking simply at a cysteine transporter. We're looking at co-transport. But the fact that we see that overexpression leads to increased glutathione and that cysteine is a rate-limiting building block, I think definitely tells us that there, for sure, we are increasing cysteine transport. It has to be. But in terms of the inhibitor, I think it's a different story, and we cannot be certain that we are inhibiting cysteine transport there. We cannot know this for sure because we also don't see a decrease in glutathione in these mice. So I think there it could be more of a glutamate effect indeed.
Given
my
this
last question
answer,
will be... Okay.
Professor Moya, I'm sorry. Yeah, that's
fine.
That's why we have a reception afterwards. Quite a lot of questions go on. Yeah, thank you very much for your contribution. Much appreciated. We go on with the next opponent, Dr. Gerlach. He is a neurologist from the Academic MS Center Zuid and Maastricht University. Welcome, and the floor is yours.
Hi. First of all, I also want to congratulate you with this nice thesis, really beautiful. Also the introduction, quite clear. And I would like to congratulate the supervisors, co-supervisors with this great work. Great job. I have a couple of different kind of questions. Maybe we'll start with statement 10. Maybe one of the parnames can read it.
Schick is mich dat.
Yeah, that's all. Could you please tell us why you choose this statement?
Esteemed opponent, thank you very much for your kind words and your nice question. Of course, I'd like to explain this. This project has been a roller coaster, and I could have said "Schick is mich dat" on multiple occasions during this project, and I did as well. A lot of experiments that completely went the opposite way, and now it kind of makes sense with the in vivo results that I've showed you here today. So yeah, it's a reminder for me to also laugh about these things and that there is always a bright side, of course.
Thank you. Yeah, I thought you would give another question. That's because of the song it's about. And in the middle of the song it says, "Zit mich deftig te eten, een vlieg in mijn vreten." And that means something like there's a bug in my food. And bugs are quite interesting insects. If you look back, they are maybe 600 million years old. And 400 million years ago, the bugs went one direction in the evolution and the vertebrata the other direction. But still, both have the same system, the eat system, somehow. It's not completely the same, but they have the same. So bugs are quite interesting. And then I look to your title. It says, all you can eat. Should we eat bugs? Could it prevent MS or cure MS?
That's a very interesting question. I've never thought about it. Perhaps, we can learn for sure from these animals, I would say. They're highly conserved, as you said. If we should eat them, I don't know if that's for everyone, but who knows, they're high protein content for sure. So, yeah,
indeed, they have some essential amino acids, like cysteine.
Yeah.
And we heard there's also a trial going on. Do you think eating those kind of substances will penetrate the brain? Since the eat system is in the brain and not at least your project.
Yeah, that's it depends on the molecule, of course, and the size of the molecule. BBB penetration is something very important for any treatments that need to end up in the brain and treat any neurodegenerative disorders. So it very much depends on what it is. In terms of when we look at N-acetylcysteine, it's fairly small. So as far as I know, it does. But for example, glutathione is slightly larger. It's 3 amino acids combined, so that gets more difficult. And some bioavailability issues there as well. So it depends on the size of the molecule and what you want to obtain, of course. But yeah, it's an interesting thing to study, as well as we did also in my thesis. It's not trivial to just say, okay, a compound ends up in the brain. And it's something that you have to, I think, study as early as possible in your research if you want to use something as a treatment for a neurodegenerative disorder.
Yeah, and that brings me to chapter 5. I agree, of course. And in chapter 5, you describe the way how academic persons should evolve and get a medicine eventually. But there's one part that wasn't in this chapter, and that is the search for brain penetration and side effects. Since if I see patients, a lot of patients use medication, but also have side effects, and that's often the reason why they don't take it anymore. So do you think there's a way to look at this product like you did earlier to side effects?
For sure, I think it's, well, of course, in a dish model or in iPSCs, you cannot really see if there's any side effects present. So I think you still need in vivo models for this. And I think there is just really important that you watch your mice and you really observe your mice, do proper tests, do behavior tests. We tend to do in our lab quite a lot of behavior testing on our mice because we think it's important. I think also there, for example, in my studies, I've done a Y-maze test. It's not in a thesis because it didn't work because these mice were behaving strangely. And I think that's also a sign. I cannot pinpoint it to a certain side effect that might translate to humans, but it's definitely something I think to keep in mind, that you see this certain behavior in your mouse, you are aware of it, and you need to then carry this with you in the rest of your research as well, and make sure that you are at least alert to these things, and make sure indeed that any side effects are spotted as early as possible, as well as toxicity, of course, which is more dramatic.
The E3 system is also in the liver and the kidneys, I guess, so you could expect side effects from that point of view. Is it possible to test the mouse liver and kidney functions and see if it's...
For sure, yeah. I think you can easily isolate those organs as well when you have your sacrifice time points and do basic stainings on the structure, as well as take along simple in vitro models with these cell types and just throw on your compound and see what happens, and see simply if your cells survive, how they act. I think that's definitely something, if this project would progress further, that you need to take into account, because otherwise it's no use to any human, I would say.
I totally agree.
Perfect timing, I would say. Lieve van Veggel, the time has passed for defense. We will now ask you to wait here while we as a committee deliberate the quality of your thesis and the quality of your defense, and leave your audience wondering whether there will be bugs on the menu in the reception afterwards. We will see what happens. I adjourn this session.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. by now, don't waste all your time. 'Cause I'll go the extra mile.
Lieve van Veggel, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor van Mierlo is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible? By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Lieve van Veggel, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affix it with the official seal of the university. Namens het onderzoeksinstituut Mens ben ik tevens gemachtigd om het officiële certificaat uit te reiken. De eigenaar van het certificaat heeft aangetoond een brede opleiding in academische vaardigheden te hebben ontvangen, een relevante ervaring te hebben opgedaan en die bijdraagt aan een succesvolle loopbaan als zelfstandig onderzoeker.
Namens Euron, de European Graduate School of Neuroscience, ben ik tevens gemachtigd om u het Euron-certificaat uit te reiken. Euron is een netwerk van 8 universiteiten in Nederland, België, Duitsland, Frankrijk en Luxemburg. Het certificaat wordt toegekend aan gepromoveerde onderzoekers die deelnamen aan meerdere internationale cursussen en conferenties van het Euron-programma, die aan de buitenlandse onderzoeksinstellingen hebben gewerkt en die hun werk hebben gepubliceerd in internationale wetenschappelijke tijdschriften. Alsjeblieft.
Beste Lieve, beste dokter Van Veggel, het is me een eer om jou als eerste te mogen feliciteren met je doctoraat. Gefeliciteerd. Je hebt het gehaald. En hoe? Je hebt vandaag in een erg heldere presentatie getoond wat je de afgelopen jaren onderzocht hebt. De jury was er unaniem lovend over. Ook de manier waarop je de vragen hebt geantwoord en vanuit de diverse jurystandpunten getuigt van een enorme expertise die je hebt verworven. Ook wil ik je familie betrekken in de felicitaties. Je man Omar, je ouders, Lou en Anja, je zus Joske en de hele familie, want ze hebben allemaal als een team achter jou gestaan. Dat hebben we zelf ook mogen ervaren op de verschillende Nicer en Rice retreats die we bij je ouderlijk huis hebben mogen houden. En samen met de bijzondere kabouters en het hart van je vader op beeldscherm gaan die mij zeker lang bijblijven. Verder denk ik ook terug aan je geweldig trouwfeest en de nuchtere inzichten waar we met het hele team op konden terugvallen vanuit Omar. Dus dank je ook, Omar. Maar ik zou ook even willen terugblikken. Zeggen en schrijven 21 mei 2019 kreeg ik een e-mail van collega Schreiber vanuit de faculteit Psychologie. En Rudy informeerde me in die e-mail dat hij een interessant druggable target had en of we hier eventueel eens over zouden kunnen brainstormen over de context in multiple sclerose. En hij zei er ook bij, ik heb 2 mogelijke zeer goede kandidaten die op dit project zouden willen starten. En al in dat eerste gesprek dat we hebben gehad, ik herinner het mij nog heel goed, met ons vieren, was ik al overtuigd dat jij een heel geschikt kandidaat zou zijn voor dit project. Je kwam zeer vastberaden over en heel even heb ik gedacht, can I manage this? Maar dat bleek al snel een unieke onderbouwde, constructieve en rustige manier van vastberadenheid te zijn die je bezat. Dus zo gedaan, zo afgesproken, zo gedaan. Jij zou dus een senior internship starten bij ons in het team om out of the box te onderzoeken of I3 een targets in MS zou kunnen zijn. In ieder geval werd je succesvol aan Melissa geklikt. We hebben al snel veelbelovende preliminaire data gegenereerd en we beslisten om voor een PhD-grant aanvraag te gaan. Je nam hier een vlotte lead en we kwamen tot een high-risk, high-gain, domeinoverschrijdend onderzoeksproject. En je hebt het project geweldig getrokken, geschreven en modeling verdedigd en je hebt hier ook verschillende grants voor gekregen om het project uit te voeren. We hadden er terecht alle vertrouwen in dat jij het project succesvol tot een einde zou brengen. Jouw chemische en moleculaire neuroscience-achtergrond waren een recept om succesvol, bijna worry-free te gaan koken. Dacht ik. Maar nog voordat het project kon starten, COVID. Het lab op slot. Maar je liet je niet uit je lood slaan. En ik was al heel blij dat je heel swift actie ondernam en besloten hebt om een hele uitgebreide review te schrijven, samen met Jan. En dat die review succesvol is, blijkt ook uit het feit dat hij al meer dan 100 keer geciteerd is inmiddels. Lieve, eerlijk waar, ik ken niemand die zo efficiënt en pragmatisch kan omgaan met Jan. Ik bedoel met het veranderende omstandigheden. Je bent meermaals zwaar wetenschappelijk op de proef gesteld. Ik kan een eindeloze lijst maken van dingen die niet liepen zoals gepland. Van de genoemde genetische gemodificeerde dieren uit Chili die op een vliegtuig moesten in coronatijd, tot cruciaal falende infrastructuur op belangrijke momenten, contaminaties en zelfs... sorry Sam. En zelfs gepubliceerde biosensoren en compounds die niet deden waarvoor ze beschreven waren. Je hebt het allemaal overleefd. Je wist steeds opnieuw heel rationeel en rustig een vervolgstap te bedenken om uit die impasse te geraken. En dan hebben we het nog niet gehad over die plot twist in je thesis. Voortschrijdend inzicht. Ik heb vaak versteld gestaan van je enorme veerkracht en maturiteit. Zelfs die plot twist heb je weten ombuigen en het neerschrijven van heel toepasselijke perspectief. Een andere kwaliteit van jou is netwerken. Ik heb veel bijgeleerd van de enorme efficiënte manier van hoe jij heel betrouwbaar je netwerk hebt gebouwd. Vele mensen weten het niet, maar dit is niet alleen een double degree tussen 2 universiteiten, maar ook 4 faculteiten zijn vertegenwoordigd, 4 verschillende faculteiten zijn vertegenwoordigd in het promotieteam. En Lieve heeft het team samen met haar netwerk samen uitgebouwd en ze heeft hier altijd de lead ingenomen. Zo is ze ook in contact gegaan met professor Mooyen, met professor Heitman, alle experimenten zelf ter plekke ook gaan uitvoeren en dat is niet vanzelfsprekend. Lieve, dit onderstreept ook wie je bent. Je bent niet alleen een warm persoon, maar ook iemand die dingen aanpakt en oplost. Hoeveel stappen die oplossing ook mag kosten, je blijft altijd rustig en je pakt door. Je nieuwe werkgever mag erg blij zijn met jou in het team. Want jij bent een meerwaarde voor elk team. Het was voor mij ook een fantastische ervaring om je te zien groeien als PhD-student. En ik wens je superveel succes en hoop je nog te zien op de volgende teambarbecue. We gaan je missen in het team. Ik geef nog even het woord aan de collega Schouder.
Well, I prepared this in English. I'm not going to switch back again to Dutch. Well, it's a moment of reflection, isn't it? It's the end of the journey. And when I was thinking about this journey, I was thinking about one of the educational elements in the research master that you also attended, and that was the pitch training. And in the pitch training, we are using a narrative framework. It's called the hero's journey. We all know that from Hollywood plots, right? It's 3 components. There's the adventure, the transformation, and then the return. But it all starts with the ordinary world. Back then, the research master, which was for some students challenging, but for you, I guess, ordinary. And then, of course, the call to adventure being the second canonical step in the hero's journey. And that was, of course, the PhD. And thinking about it, the hero's journey is not only for you, but I think it's for a lot of PhDs. But some journeys are a bit more challenging. And as we just heard today, this one qualifies for that as well. And then the tests, the allies and enemies. I'm not sure you made enemies, but you certainly had the tests. We have seen them, we have heard about them. It's reflected in your statements number 10 and 12 as well. And it culminates in the ordeal. This described as the central crisis transformation. I'm not really sure you had that because it seems to be more a constant crisis. I'm thinking about modern shows like Breaking Bad, where the main characters are never safe. But then there is the reward. And I had to think about, your thesis, the light, and us sitting here today. And this brings me what I think is the most intriguing component of this hero's journey, the return with elixir. I translate elixir as toverdrank, and I just paraphrase what is meant by that. The hero returns to the ordinary world, now transformed, and brings back something of value, by this knowledge, physical object, or new understanding. This elixir benefits both the hero and their community. So I leave it by this cliffhanger for everyone to reflect, including Lieve. What is the elixir that you're going to bring from this adventure? Having said this, congratulations. You made us proud. You can be proud of yourself. I wish you all the luck with your next hero's journey, and this time as a young professional with Brightlands.
Dr. van Veggel, congratulations. Yeah, you have to get used to that, right? And the other thing is that they're the young doctor for the next few weeks. That's the official pronoun. For 6 weeks, you're the young doctor, which takes a generation away, and then you're suddenly in kindergarten, so we're in trouble. Congratulations on behalf of this university. We're very proud of you. You're an alumni of this university now forever. I give back from the deliberations that we have that people were really impressed by your presentation. It was a very nice presentation, understandable for almost everyone, I think. So well done. And also the quality of your answers and the breadth of your knowledge that you displayed was exemplary. So very well done. We will almost conclude this session, but there are a number of things you need to know. We first will stay here as a committee because we make a number of pictures, and then the people from outside, so online, can also be in the picture because they will be projected behind us. And then there will be the possibility for them to congratulate you. Then we go to the stairs, so your closest family members, or the ones that you really like, can be in that picture as well. You never know, right? There it starts. And then we will proceed for people who stay here to congratulate you here at the stairs. And the rest of the audience can already go to Tribunal. En we nemen er eentje. Hè? And then the young doctor might arrive at some time to have the rest of the congratulations. So thank you very much. Enjoy the day. And the committee here present, thank you very much as well. I close this session.